we're now going to go into cloning and transformation. This is a little bit, so we were just with DNA, and now we're kind of more into genes and potentially later protein products. So we are going to start off with molecular cloning, which the main function is to create more copies of a particular gene that we want. And so the first step for molecular cloning would be to first insert the sequence of interest into a plasmid, specifically with restriction enzymes. And like I mentioned earlier, restriction enzymes have palindromic sequences, and this is how I'm going to show it a little bit more. But palindromic sequences basically means that the 5 to 3' would be the same both ways, even if you were to do a complementary sequence for this. So as you can see, we have the 5' to 3' here for G, G, A, T, C, C. And then if you were to make that complementary sequence, so you would start off with 5' here, and then the complementary base would be a G, another G, an A, and T, and then a C and a C. So as you can see, and then it ends with a 3'. So as you can see, the sequences are the same, even though it's this one and then the complementary sequence. And these restriction enzymes create a break on each DNA strand. And so at this bottom portion here, I have it sort of visualized. It goes through a little bit more in depth than what I'm mentioning here, but for the purposes of the MCAT, it doesn't go too crazy in depth. So these are just some like an outline of how the process goes. But so yeah, it creates the 2 breaks on each of the DNA strands. So then once we get there, we have these 2 sticky single-stranded regions so once you put the break, they can either rebind via hydrogen bonding to their complementary sequences, or they can also bind to the same complementary sequences elsewhere if you have another strand somewhere else that also has a complementary sequence. So that's in the case that we have these 2 sticky single-stranded regions. And then in the case that the same palindromic sequences are placed on both ends of the cDNA and the plasmid, because as you know, in the first step, we're trying to insert the sequence of interest into the plasmid. But if both of them are already kind of have that break, then the sticky ends of the cDNA can bind to those of the plasmid. And this is kind of what I'm in a sense illustrating here, where we have these sticky ends already of this plasmid, which is that darker red color, because the original blue one here is the original plasmid, but that's before it got cut. And then this is the other, DNA that we also cut in order to kind of match into here. And then, so this is another way that could happen. And then in order to create more copies of this gene, we have this new plasmid with that specific DNA sequence. And then we add this plasmid to a bacterial colony, and then through transformation, the bacteria will be able to incorporate that cDNA and the other genes that are on that plasmid into its genome. So then it creates more copies of that gene as the bacteria continues to replicate. And also many common ways to test that the insertion, and then also that the bacteria is creating these genes from the plasmid that we aim to have, is to add an antibiotic resistance gene into the plasmid. And so as the bacteria is, creating that antibiotic resistance gene, we add that antibiotic in there and see if the bacteria is actually showing that property of being resistant to the antibiotic. Or also there's a reporter gene that we can add, which usually creates either a specific type of protein or a specific fluorescence, just another like reporter gene, kind of similar to the probes that we were talking about before, where that can be visualized to see if the incorporation of that gene into the plasmid was successful. And so I mentioned over here transformation, but just a reminder of what transformation is that it is when bacteria are able to incorporate the genetic information from its environment into its own genome. And so that's why when we add that plasmid to the environment of the bacteria, then it's able to take up that plasmid, incorporate it into its genome, and then produce the copies of the gene that we intend it to for molecular cloning. And also, just I realized I didn't define plasmid before, but it's a small circular piece of DNA. And then once we add that specific sequence in, then it would be containing the gene of choice that we want the bacteria